The induction of synapse formation by de novo neurotransmitter synthesis. One of the authors, Nicole Wang, says this was a fun one to be part of. Turns out you can convert glutamatergic synapses into GABAergic ones simply by expressing VGAT and a couple of GATs. You all should check it out. I agree. You all should check this out. So basically what the authors did is they used transgenics somehow to make neurons that are glutamatergic, so neurons that normally release glutamate. They gave that neuron the machinery to also release a different neurotransmitter, GABA. It's a gamma aminobutyric acid. And then they recorded from the postsynaptic neuron, the one next over. So that neuron used to get glutamatergic input. And now suddenly, the presynaptic neuron, instead of releasing glutamate, it starts to release GABA. What happens? And it turns out the postsynaptic neuron just said, okay, you're releasing GABA now, that's fine. Then I'll put some GABA receptors here, and I'll eat that GABA, and I'll start to respond to that GABA. So I don't know.
That's quite crazy.
I find it's really cool to see that. Because how would a neuron know, right? How would a neuron know? Because you need to match the neurotransmitter that's being released by one neuron with the receptor on the other neuron. Otherwise, the communication doesn't work. If I have a receptor for GABA and you're releasing glutamate, that won't bind. That won't work. I won't receive that glutamate. So something on the postsynaptic neuron must say, okay, I'm seeing a lot of GABA now. You know what? We'll make a few GABA receptors. And that is apparently how it works.
That's weird.
That is right.
Yeah, that's interesting.
So there's a whole field of synaptic adhesion proteins and scaffolding proteins and all sorts of things that kind of organize the match between the pre and the postsynaptic neuron to make sure that they work with the same neurotransmitter. And basically all you need for all of that machinery to work is just start releasing a new neurotransmitter, and the postsynaptic neuron will know what to do. Which is, I don't know, I find this all sorts of cool. Obviously, because I sent this to review and accepted it.
Oh, you're promoting your favorite submission of the month.
I like this work. It's nice to see it out.
But I have a question. So the synapses that they made GABAergic, the presynaptic neuron, it also kept releasing glutamate.
As far as I understand, yes. I don't remember the details.
Okay. So they did the genetic, some transgenetic thing to make it both.
Mm-hmm.
It doesn't delete the glutamatergic synapse. That's cool. But does the brain? They did it in mice, right? So does the brain do anything else if you do it in in the whole mouse? Did they try this, or did they do it very locally and then nothing horrible?
So they limited this specifically to purely glutamatergic neurons. I don't think that affected enough neurons to really see a difference in behavior.
Yeah, that could be. Okay.
I don't actually know.
I was wondering if you make suddenly a lot of glutamatergic synapses, GABAergic and glutamatergic. I was wondering if something changes, or maybe nothing changes because the connectivity is robust and you just let the mouse live its life, and then it's so plastic that it actually fixes it itself. I don't know.
That's possible. Right, so this was a study by Scott Burlingham, Nicole Wong, Lindsay Peterkin, Lily Lubo, Carolina dos Santos Passos, Orion Banner, Michael Gabriel, Thomas Kast, Matthew Friedman, Xiu Friedman, Thomas Sudhoff, and Soham Chanda. Now out in Nature Communications.
Very cool.
Right.